Okay, let's unpack this. Today we're doing a deep dive into ion channels. These tiny molecular machines, they basically define electrical signaling in, well, every cell in your body. Our aim here is pretty straightforward. Cut through the complexity, get to the core of their structure, how they work, and interestingly, why they matter so much for medicine. First off, ion channels are literally everywhere. Pore-forming proteins stuck in cell membranes, and the speed is just stunning. We're often talking over 1 million ions shooting through per second.
Right, and that speed, that sheer efficiency, it hinges on a really critical energy rule. You see, unlike a lot of other cellular transporters, ions move passively. They strictly flow down their electrochemical gradient.
Right.
Downhill, you could say. No metabolic fuel like ATP is burned in the process.
Okay, electrochemical gradient. That sounds a bit technical. Can you break that down simply for us listening?
Absolutely. Think of it like this. Ions feel two main poles across the membrane. There's the chemical force, just the difference in how many ions are inside versus outside, simple concentration. And then there's the electrical force stemming from the ion's charge and the voltage difference across that membrane. It's the combination of these two natural forces that drives the movement. And this passive flow is the only way biology can achieve those, frankly, breakneck speeds needed for things like nerve signals. Active pumping just couldn't keep pace.
So they're really the bedrock of biological speed then.
Yeah.
Essential for the nervous system, obviously, but you mentioned they're not just in the brain.
Oh, not at all. They're the basis of the nerve impulse, yes, and signaling across synapses. But they're also key wherever you need really rapid, coordinated cell responses. Think about the precise timing needed for your heart muscle to contract, or how quickly insulin needs to be released from your pancreas. Ion channels are running the show there too, and their structure is quite elegant for this. They're integral membrane proteins, usually several subunits coming together, forming this water-filled pore right through the membrane.
Hang on, if it's just a pore, how does the cell control what gets through? I mean, the notes mention the narrowest point is maybe just 1 or 2 atoms wide. That seems less like a superhighway for 1 million ions a second, and more like a tiny bottleneck, doesn't it?
Ah, that's the beautiful paradox, isn't it? Speed and specificity. The magic happens in what we call the selectivity filter. It was scientists like Bertil Hill and Clay Armstrong who first figured out the concept. They proposed that this filter region, it's lined with specific atoms from the protein itself, carbonyl oxygens. These oxygens are perfectly positioned to essentially mimic the water molecules that normally surround an ion, say a potassium ion.
Yeah.
They effectively swap places with the water, letting the potassium ion slip through smoothly. But a smaller ion, like sodium, even though it's smaller, it holds onto its water molecules more tightly in that specific chemical environment. It can't make that perfect fit with the filter's oxygen, so it gets excluded. It's incredibly precise.
Oh, okay. So an atomic level bouncer, basically. That sounds almost theoretical, something hard to actually see. But didn't someone manage to prove this structurally?
Exactly. And that's where MacKinnon's work came in. Using X-ray crystallography, he determined the actual molecular structure of a bacterial potassium channel, KCSA. And boom, there it was. The structure perfectly matched the Hill and Armstrong model, showing those carbonyl oxygens lining the pore. That work earned him a share of the Nobel Prize in Chemistry back in 2003. It was a huge confirmation.
Okay, so we know how they filter, but what about control? How does the cell decide when to open or close these gates? What flips the switch?
Right, gating, that's the term. It really depends on what kind of stimulus the channel is built to respond to. 2 main types dominate. You have voltage-gated channels. These guys open or close based on the electrical voltage difference across the membrane itself. Think of the sodium channels crucial for firing off a nerve impulse and action potential. Then there are ligand-gated channels. These need a specific chemical molecule, a ligand, often a neurotransmitter, to bind to them, usually on the outside of the cell, to trigger opening. A classic example is the GABA receptor in the brain. GABA binds, the channel opens to chloride ions, and this inhibits nerve activity. Actually, many anti-anxiety drugs target this very channel.
Which leads perfectly into pharmacology. Because they're such precise gatekeepers, it makes sense they'd be major drug targets.
Yeah.
Both natural toxins and clinical medicines seem to exploit them. You see these incredibly potent blockers in nature, like tetrodotoxin, TTX, from pufferfish, right? Or saxitoxin from certain algae. Nasty stuff because they just completely jam sodium channels.
Absolutely potent and often irreversible. They taught us a lot about channel function. And yes, we use that same principle blocking channels in medicine, but thankfully in a much more controlled and reversible way. Think about local anesthetics, a lidocaine, novocaine. What
Yeah.
they do is temporarily block those same voltage-gated sodium channels, but only in the local area where they're applied. This stops the pain signal from traveling up the nerve. No signal, no pain perception in the brain. Simple, but effective.
But when this precision engineering goes wrong naturally due to genetics, we get diseases, right? These channelopathies. Cystic fibrosis is probably the most recognized example.
That's correct. Cystic fibrosis is a devastating example. It's caused by mutations in the gene for a specific chloride ion channel called CFTR. When that channel doesn't work properly, chloride transport is disrupted, and this throws off the balance of water movement across cell membranes, particularly in the lungs and digestive system. That leads to the thick, sticky mucus characteristic of the disease, a single-channel defect with massive systemic consequences. And just to give you a sense of the sheer scale and diversity here, we know of over 300 different types of ion channels just in the cells of your inner ear. 300. Just for hearing and balance.
Wow, that number is,
Yeah.
quite humbling. Millions of ions per second through pores, atoms wide, controlled with such precision, hundreds of types.
Yeah.
All just humming along, keeping us alive.
And here's maybe a final thought to leave you with. For all this incredible biological complexity, this intricate molecular machinery, the way these channels flicker between their different states, open, closed, sometimes inactive it turns out, their overall behavior, averaged over time, can often be modeled quite accurately using mathematics, specifically using things called stochastic processes like Markov chains. It's this amazing bridge where pure math meets molecular biology to predict the behavior of life's fundamental switches, something to ponder.